Assalamu alaikum everyone. Inshallah you're all doing fine. We'll be talking today about blood coagulation. Now this is a beautiful lecture, as you will see, and I hope you will enjoy it. So let's start definition of what blood coagulation is. So it is basically it describes an orchestrated biochemical process. So I chose the word orchestrated because as you read it, as you go through it, it's almost like a music. It's like a classical music of Chopin or Beethoven, or Mozart or whomever. You will see that it's like a classical music. So it goes slow and then it goes up and up and up and it reaches a climax, and then you see the music going down until you get to the end. So it's really basically a symphony. So it's basically, again, it's a biochemical process. It is initiated as a result of vascular injury. So you have an injury with in a small area, and blood in this area changes from liquid to gel. So basically you have the formation of a clot of fibers of a protein known as fibrin. Now that results in hemostasis, so there is no more blood loss, and then this is followed by clot, dissolution, and repair. That is the end of the symphony. These are the basic steps. You have a physiological effect first. Basically, you have vascular constriction. It limits blood flow to the area of injury. This is followed by a chemical process, cellular and biochemical. You have activation of platelets and their aggregation at the site of injury, forming what is known as a platelet plug. Now this platelet plug is loose, and it becomes solidified and it becomes hard as what we call a hard clot via the formation of a fibrin mesh. So you have a network of all of these fibers surrounding and entrapping the platelet cells. Now this is followed by, again, dissolution of the clot enzymatically, as we will see at the end of this lecture. So what are platelets? Platelets are small anuclear cell fragments. They are produced from large cells known as megakaryocytes. Okay, so you have these cellular fragments that are produced from these cells. Now inside platelets you have cellular components. You have, of course, the plasma membrane, but you also have the actin cytoskeleton. There are vesicles, there are proteins, there are signaling factors, and we will talk about all of these. Now, inside these platelets, there are granules or vesicles. Now, there are three types of vesicles. You have electron-dense vesicles. These contain calcium ions. They also contain ADP, ATP, and serotonin. Now, the reason why you have ADP and ATP inside these vesicles is that they're not really used as sources of energy. Rather, they are used as signaling molecules. You have also alpha granules inside platelets, and these granules contain heparin antagonist, the signaling molecule platelet-derived growth factor. You have the structural protein fibrinogen, a regulatory protein known as von Willebrand factor, and there are numerous clotting factors inside these vesicles. And then you have lysosomal granules or vesicles, and these contain hydrolytic enzymes. Now these enzymes are necessary again for removal of the clot, activation of different proteins, and so on. So what happens with these platelets is that once they're activated, so once there's an injury and once they're activated, these granules fuse with the plasma membrane, releasing their contents. So here you have a platelet, a representation of a platelet, and you can see there are numerous receptors on the cell surface. You have a receptor for epinephrine, a receptor for ADP, another for thrombin, and so on. There are also glycoproteins on the cell surface, and these are important for interacting with collagen, van Willebrand factor, fibrinogen, and so on. These are also important for forming aggregation of platelets at the site of injury. Now, also on the surface, you have the process of blood coagulation going on. Okay. Now, so first thing that happens is that whenever you have a vascular injury, a protein known as van Willebrand factor is exposed. Now, this leads to platelet binding to this van Willebrand factor and activation of the platelets. That would cause a series of signaling reactions inside the cell, and that leads to secretion of a number of factors. Again, you have ADP, serotonin, factor V, calcium ions, ATP, fibrinogen, more van Willebrand factor, thrombin, and thromboxanes. Now, what happens? These bind to receptors, or they have specific functions, as you will see. Now, what happens as well is that once the platelets are activated, their shape changes. Now, this change in cell shape results in more platelet adhesion and aggregation. Now, one of the receptors that exists on the surface of platelets is a thrombin receptor. Now, thrombin receptor activates a G protein that eventually activates the enzyme phospholipase C gamma. Now, all of these receptors, by the way, activate G proteins. So what happens here is that phospholipase C, once it is activated, it can act on its substrate, phosphatidyl 4,5-bisphosphate, PIP2, and it leads PLC, phospholipase C, degrades PIP2 into two products, and these are known as the diacylglycerol and the phosphatidyl inositol triphosphate. Now, what happens with IP3 is that it induces the release of calcium ions from stores like the endoplasmic reticulum. Now, diacylglycerol activates protein kinase C. So let's focus now on calcium ions and what they do. So basically what they do is that they can act on plasma membranes, and they activate a phospholipase enzyme known as phospholipase A2, and this enzyme releases arachidonate from phospholipids in the plasma membrane. Now, arachidonate can be converted by cyclooxygenase and then thromboxane synthase all the way to formation of thromboxane. Now, thromboxane can be released, and once released, it has vasoconstrictor activities, again limiting blood flow and helping in the aggregation of platelets, and it can also act in autocrine and paracrine manners. So what thromboxane does is that it can bind to its receptor on the cell surface on the same cell that secretes it, activating signal transduction pathway inside. It can also act on neighboring platelets as well. Now, we talked about this in the introductory biochemistry course when we talked about enzymes. We talked about lipids, if you remember, and thromboxanes and prostaglandins and arachidonate are eicosanoids. They are 20-carbon fatty acids. So arachidonate is converted to prostaglandins by an enzyme known as cyclooxygenase 1. Cyclooxygenase 1 is a target for aspirin. So what this happens, this is why it has always been thought that aspirin is beneficial, because it prevents in the formation of atherosclerosis and aggregates of platelets. So this is the mechanism of action of the non-steroidal anti-inflammatory drugs. Now, some other molecules that are secreted as well is serotonin, which is also a vasoconstrictor working alongside thromboxanes, and the platelet-derived growth factor, which stimulates proliferation of endothelial cells, and this also reduces blood flow. So what calcium ions do, in addition to activation of phospholipase A2 and release of arachidonate from phospholipids, is that it can bind to a kinase known as myosin light chain kinase. It phosphorylates myosin light chain, and this phosphorylated myosin light chain has a number of effects. One of them is that it stimulates the further fusion of these granules with the plasma membrane, and eventually they release their contents. Now, myosin light chain kinase also modifies the actin cytoskeleton, so it can modulate, it can induce motility, and it also changes cell morphology, platelet morphology, leading again to more aggregation of platelets. Now, the other side of the equation is diacylglycerol, which is the second product of hydrolysis of PIP2. What diacylglycerol does is that it binds to protein kinase C. It phosphorylates several proteins, including protein 47, and this leads to, again, further release of granular contents, including ADP. So what does ADP do? ADP is a platelet activator. What it does is that it binds to its receptor, all right, and it modifies platelet membrane, allowing for fibrinogen to bind to platelets, to the glycoproteins on platelets, resulting in further platelet aggregation. And this is known as a platelet plug. And we'll talk about platelet plug later on in this lecture. Now, something else is that the surface of platelets is the theater for the blood coagulation process, the biochemical reactions that lead to blood coagulation. And this is the topic, or the next topic of this lecture. So let's talk about coagulation, the biochemical reactions that take place in order to form blood clots. Now, there are a number of players in the process of blood coagulation. Some are small, like calcium ions and vitamin K. Others are large, like platelets themselves. And you have molecules in between. So first, we have the organizing surface of platelets. We already talked about that. You have also proteolytic zymogens. That is, remember what zymogens are. Zymogens are enzymes that require proteolytic cleavage in order for them to be active. So in introductory biochemistry, we talked about trypsinogen and chymotrypsinogen, proelastase. They require chemical modification, proteolytic modification, generating the active enzymes, trypsin, chymotrypsin, elastase, and so on. Now, these zymogens in the process of blood coagulation include pre-kallikrein, prothrombin, as well as a number of proteins known as factors, and they are designated with Roman numbers, and these are known as factors 7, you have 9, 10, 11, 12, and 13. Now, these are mainly serine proteases. They are released from hepatocytes. Once they're activated, they are designated with the subscript A. So, for example, factor 13, once activated, is called 13A. You also have other proteins that function as anticoagulants. So they are inhibitors of blood coagulation, and these include protein C and protein S. There are non-enzymatic protein cofactors, including factors 8, 5, and a protein known as tissue factor. As I said, there are calcium ions, vitamin K, and then we have fibrinogen, which forms the fibrin network. So this is a list of the different clotting factors, their names, and what their functions are. So as you go through this lecture, you can go back to this slide to sort of like summarize what the process is. So note that factor 2, for example, is the prothrombin. Factor 1 is fibrinogen. Okay. Factor 4 is calcium ions themselves. Okay. So these factors do not necessarily indicate a protein. Now classically, blood coagulation has been classified into two pathways that were independent. They were thought to be independent. And these are known as the extrinsic pathway, and the other is the intrinsic pathway. So basically, the extrinsic pathway is activated in response to tissue injury. The intrinsic pathway can be activated as a result of an internal effect like inflammation, for example. So basically, but it has been found that actually these pathways are not totally independent. Rather, there is a bridge that connects both to each other. And when the extrinsic pathway is activated, you have activation of the intrinsic pathway as well. So they are really interconnected and not totally independent of each other. But both pathways converge at a single point, which is activation of factor 10. And factor 10 is responsible for activating factor 2, which is thrombin. And thrombin can form, again, the fibrin polymers or the fibrin mesh or network. So what we will do is go over this pathway right here. Now, so you have the extrinsic pathway. You have vascular injury. Something happens to the vasculature. Exposure of collagen as well as von Willebrand factor result in the activation of factor 7. Factor 7 then, with the help of tissue factor, it activates factor 10. Now, in the intrinsic pathway, the tissue factor also plays an important role as it forms a bridge whereby whereby factor 7 can activate factor 9, and factor 9 can activate factor 10. So as I said, you have both pathways converge at this point right here with the activation of factor 10. Now factor 10 activates prothrombin. Thrombin forms the fibrin network. Now, in case of inflammation, you also have activation of the kallikrein-kinin pathway, where I have activation of factor 12, which activates factor 11, and factor 11 activates factor 9. So there are a number of cofactors that play an important role in this process. And these are the non-enzymatic cofactors like tissue factor, factor 8, factor 5, and so on. Now, pay attention also to the involvement of phospholipids, that is the surface of platelets and calcium ions, in the process of activation. Now, so this is sort of like an illustration of the different domains, the different proteins and different domains. Now, what I care about really is the presence of a domain known as the GLA domain, or the glutamate domain right here. And this glutamate domain, if you notice, it exists in the prothrombin protein, factor 7, factor 9, factor 10, as well as protein C and protein S, the anticoagulants. So what is the GLA domain, or the glutamate domain? So basically, this domain is a sequence of it's part of the primary structure of these proteins, factors 9, 10, 7, and prothrombin, and the other ones that I just mentioned. Now, in this domain right here, it's rich with glutamate. So you have about 9 to 12 glutamate residues within this domain. This is actually, glutamate residues are the substrates for an enzyme known as a carboxylase enzyme. What this enzyme does is that it adds another carboxyl group. So you have a glutamate with 2 carboxyl groups, looking like this. That leads to interaction between glutamate with the calcium ions. Okay. Now, and this helps in binding of these proteins with the plasma membrane of platelets. So you have in the plasma membrane, you have the phospholipid head groups, which are negatively charged, and calcium ions are positively charged. So calcium ions sort of mediate interaction of these proteins with the plasma membrane. The interaction or the insertion of these proteins with the plasma membrane is also solidified by having a hydrophobic region in these proteins. So that helps inserting the proteins into the plasma membrane. And that's what makes the coagulation by a chemical process take place on the surface of platelets. Now, the carboxylation reaction requires vitamin K as a source of electrons. So vitamin K is oxidized into what is known as a vitamin K epoxide. Now, and you have the carboxylation reaction taking place, addition of a carboxyl group to glutamate. Now, it's very important for vitamin K epoxide to be reduced to regenerate the active form of vitamin K, and this happens also enzymatically with an enzyme known as vitamin K epoxide reductase enzyme, and this requires NADH as a source of electrons. This enzyme right here is the target of the antagonist, famous antagonist known as warfarin. and we'll talk about it later on. A side note on vitamin K is that newborns are at risk for early vitamin K deficiency, and that may lead to bleeding. The reason is that there are 4 reasons actually. The first is that the placenta really is a poor passage channel for fat-soluble compounds such as vitamin K. Neonates are also born with immature liver that is not able to produce these coagulation factors and to modify the factors at the glutamate residues. Something else is that breast milk is a poor source of vitamin K, and the last reason is that the intestinal flora, which are the main source of vitamin K in humans, is not really mature yet. It's not established. Now, how do we know that these newborns have vitamin K deficiency? There are a number of symptoms, or the number of signs or manifestations. One of them is bleeding, gum bleeding, as well as formation of bruises and so on. So what happens is, as I said, the 2 pathways converge at the activation of factor 10. So factor 10 forms a complex with factor 5, and this is known as the prothrombinase complex. This interaction takes place as a result of interacting with the calcium ions. Same thing happens with prothrombin. It can also interact with factor 5 with the help of calcium ions. Now, so factor 5, what it does is that it brings both of these, factor 10 and prothrombin, close to each other, and factor 10, which is already active, it cleaves prothrombin, forming the active enzyme, thrombin. So we have the involvement of other factors in the extrinsic pathway and the intrinsic pathway as well. So in the extrinsic pathway, prior to activation of factor 10, you have the activation of factor 7 with the help of tissue factor. In the intrinsic pathway, you also have the involvement of tissue factor, which activates factor 9, and it can activate, or it can help in the activation of factor 10 by associating with factor 8. So we have factors 5 and 8. So both of these are cofactors that increase the proteolytic efficiency of factors 10 and 11. Okay? Now, the complex of factor 10 and 8 is known as the tennis complex. So this one right here is known as the tennis complex. So what happens is that factors 5 and 3 are activated by thrombin, by the way, which comes downstream, right? So we'll talk about this as well in the amplification slide. So what happens is that factor 8 is already circulating in the plasma membrane, but it can bind to von Willebrand factor. Okay, so when it binds to von Willebrand factor, it gets close to the site of injury, and that increases, by the way, the half-life of factor 8, and it brings it closer to factor 10, where it can be activated. Now, there is a condition that is related to deficiency of von Willebrand factor, and it is associated with decreased plasma concentration of factor 8. Individuals with von Willebrand factor suffers from excessive bleeding and inability to coagulate blood. Now, tissue factor is an integral membrane protein that is expressed on the surface of activated monocytes and subendothelial cells. as well as other cells. So really, blood cells and platelets are not exposed to subendothelial cells unless they are injured, unless there is tissue injury. Then you can have platelets binding to tissue factor. Once you have this interaction with tissue factor, you can have the activation of factor 10 in the intrinsic pathway. In the extrinsic pathway, I'm sorry. Now, in the intrinsic pathway, you also have the involvement of tissue factor, whereby it is important for activating factor 9, which activates factor 10. Now, the interaction between, or the complex of tissue factor with factor 7 is known as the initiation complex, because you have both of these pathways, the intrinsic and extrinsic pathways, activated. What tissue factor does, by the way, is that it increases the efficiency, the proteolytic efficiency of factor 7. Now, upstream of factor 9 is you have factors 12 and 11. And you have activation of what is known as the kallikrein-kinin system. These are inflammatory molecules, and they do have an effect on the coagulation process. Such as kallikrein, when activated, it can activate factor 12, and factor 12 can activate factor 11. Now, kallikrein as well has other effects. By cleaving kininogen, producing bradykinin, it has vasodilation effect. Okay, so what happens is now the music is going up, okay? Now you have activation of all of these molecules, and then we're getting to the climax of the coagulation process. So you have activation of factor 10. Factor 10 activates prothrombin into thrombin. Thrombin functions, or it acts on fibrinogen. So what it does is that it converts fibrinogen into fibrin. How does it do that? By removing a number of peptides of the protein. Now this results in the ability of fibrin molecules to form electrostatic interactions between the head and the tails of two other molecules. So you have the head right here forming electrostatic interactions with the tails of two other fibrins, and so on. So what happens here is that you have aggregation of the fibrin molecules together, forming a clot, and this clot is known as a soft clot, because the interaction between all of these molecules is based on electrostatic interactions, which is non-covalent. So in addition to cleaving the fibrinogen into fibrin, thrombin can act on another protein, and it is known as factor 13. Factor 13 is a transglutaminase, and what it does is that it forms a covalent interaction, a cross-linking between a glutamine of one fibrin monomer to a lysine on another fibrin monomer. So you have the formation of now a hard clot. Okay, now because of the covalent interaction or bond or cross-linking between the different fibrin molecules. Now this clot not only contains the fibrin network, they also entrap platelets inside, forming what is known as a platelet plug. Now we're really getting to the climax of the symphony. What happens here is that you have activation of all of these molecules, and it's getting even amplified. The reason is that you have feedback activation. So you have the thrombin molecule acting back on all of these factors. So what it does is that it activates more factor 5, more of factor 8, more of factor 11, and it can also act on factor 7 as well. So you have more amplification, you have more activation of all of these zymogens, specifically getting to the end to the factor 10 that activates more and more prothrombin to thrombin. So thrombin is important for a number of reasons. One, it has its own receptor, right? So what it does is that it recruits platelets, it activates platelets, and it recruits them to the site of injury. It amplifies the coagulation complex. It forms the soft clot with the fibrinogen, with the fibrin monomers forming a network. It also forms the heart clot by activating factor 13. But what it does as well is that it terminates coagulation, and this is the topic of the next topic of this lecture. But it does have other actions as well. So thrombin itself can bind to what is known as protease activated receptors, and what these receptors do is that these receptors are present on the surface of number of cells, including endothelial cells, and what they do is that they can modulate, they can induce vascular remodeling, for example, angiogenesis, formation of new blood vessels, as well as inducing inflammation. So now the music is going down. So let's talk about the anti-clotting factors. So once thrombin is activated, what it does is that it binds to a protein known as thrombomodulin. This protein is present on the surface of endothelial cells. Now it brings thrombin closer to a protein known as protein C. Protein C is activated by thrombin forming activated protein C. It forms a complex with protein S, which is present on the surface of platelets. Now this complex, the complex of protein C, protein S, can degrade factors 5 and 8. So you have termination of activation of prothrombin and activation of factor 10. So we have another inhibitor, and it's known as Antithrombin III. Now this is a serine protease inhibitor, not only of thrombin, but it can also inhibit other proteases such as factor 9, 10, 11, and 12. So what antithrombin does is that it binds to heparan sulfate. Heparan sulfate is synthesized by mast cells, released, and it can bind to the surface of endothelial cells. So once antithrombin binds to heparan sulfate, the conformation of the antithrombin changes, allowing it to bind to factor to the other proteins, to factor 10, for example, and thrombin, inhibiting them. So you have this interaction that is also, of course, it's amino acid dependent or specific interaction. Now, what's important about heparin is that it is used in laboratories, in clinical laboratories, in phlebotomy tubes. So what it does is that it prevents blood clotting. So once blood is drawn, it's collected into these tubes and it stays in a liquid form. Otherwise, it coagulates immediately. So another inhibitor is tissue factor pathway inhibitor. Now, what this inhibitor does is that it can bind to factor 10, and then it can interact with the complex of tissue factor 7, inhibiting the whole thing. And that prevents the activation of factors 10 as well as factor 9. Now, the other thing about this protein, the tissue factor pathway inhibitor, is that it can also prevent the prothrombinase complex. By interacting with factor 10, it can also inhibit the activation of prothrombin to thrombin. Now, anticoagulants can also be used, and these are calcium chelators as well as vitamin K antagonists. So you already know that calcium is important for the process of coagulation. So by using chelators that absorb calcium ions, that prevents it compromises the efficiency of blood coagulation. Now, there is a drug known as warfarin, and what warfarin does is that it inhibits the vitamin K epoxide reductase, preventing the regeneration of the active form of vitamin K, the reduced form. So there would be inability of the carboxylase enzyme to carboxylate glutamate, and that again compromises interaction of proteins to the surface of platelets and inducing blood coagulation. So now the music is going down. Okay, I hope you can imagine that. So now what happens is that there is a clot, there is a reduction in bleeding, a cessation of bleeding. The clot must be removed, and there is renewal of cells as well. So now the clot must be removed. And this happens simultaneously with the activation of blood coagulation, by the way. Okay, so that things do not go out of control. And the dissolution of blood clots is basically enzymatically dependent. And this is done by a protein or a protease known as plasmin. Plasmin is a serine protease. It is activated from its precursor plasminogen. Now, plasmin is responsible for fibrinolysis. It's responsible for hydrolyzing and degrading the fibrin network. And it just happens that plasminogen has high affinity for fibrin clots, so otherwise it doesn't really bind to fibrin. So look at this. So plasmin is activated, it can degrade fibrin. This activation process is induced by a plasminogen activator known as tissue plasminogen activator, and it's also induced by what is known as urokinase plasminogen activator. Now, the tissue plasminogen activator is activated by protein C, the activated protein C, the anticoagulant that we talked about before that degrades factors 5 and 7. So at the same time, activated protein C inactivates the inhibitor of plasminogen activator. So what you have in here is induction, stimulation of the fibrinolysis process. You also have an inhibitor known as antiplasmin, and this antiplasmin keeps things in check. It inhibits plasmin, but not when plasminogen plasmin are clot bound. Okay, so this is beautiful. So if you have a plasmin that is soluble, that is outside the clot, it is immediately inhibited by the antiplasmin. But as long as it is bound to the clot, degrading it, lysing it, removing it, it's active and it cannot be inhibited by the antiplasmin. That ensures the removal of the clot without compromising or affecting the surrounding tissue and surrounding proteins. Something else is that there is a molecule known as streptokinase, and this is not an enzyme, by the way. Streptokinase is used clinically. It's produced from streptococci bacteria, and it can activate the circulating plasminogen to form plasmin. So what it does is that it facilitates the removal of the blood clot. So in addition to tissue plasminogen activator, there is urokinase plasminogen activator, which is again it activates plasminogen to plasmin, and it can also be used clinically. And the symphony is completed. That's the end of the music. You have initiation, you have formation of a clot, you have amplification, that's the climax, and then you have clot dissolution, end of blood coagulation, and blood clotting. I don't know who the maestro is. Is it factor 10? Is it thrombin? I don't know. You tell me. Allah ma'akum.